My name is Oliver Fien. I'm director of the West Coast Metabolomics Center, and I welcome everyone who joined us here remotely. And it is my great pleasure to introduce Professor Nadja Cech of the University of North Carolina at Greensboro. Professor Cech is a distinguished professor of chemistry funded by Patricia A. Sullivan Fellowship, I guess. She earned her bachelor degree in chemistry from Southern Oregon University at the age of 19. So really early starter, and that degree, that bachelor, was funded in part by a large crop of the medicinal plant Echinacea, which she helped to cultivate on her family's organic farm. So Echinacea is a genus of flowering plants in the daisy family and the common ingredient in cold medicines. And promoters of Echinacea believe that the supplement boosts the immune system and can reduce symptoms of infections and other illnesses, including the common cold. So I guess that was a real starter kit for her. And she followed that trajectory ever since, I understand. In 2001, she got her PhD in analytic chemistry from University of Mexico, where she already had worked with mass spectrometry and published on fundamentals of electrospray ionization, mass spec. She joined the faculty of UNC Greensboro in 2001, so for 20 years she's been there, and still applies her theoretical studies to medicinal plants. In particularly, she's interested in, obviously, metabolomics as a tool of discovering bioactive constituents of botanicals. She was awarded the Jack Beale Award from the Journal of Natural Products for the best paper of a young investigator in 2011, recognized for teaching with the 2008 UNC Teaching Award, as well as for the 2017 Thomas Undergrad Research Award, and in research for a 2011 Junior Research Excellence Award. So she has published many papers so far, over 6,000 citations in Google Scholar. And we are really looking forward to, see, a culmination of all that work today in the seminar. Thank you for joining us, Nadja.
Thank you so much. Dr. Fien, it is a real pleasure to meet you, and I have to commend you on pronouncing my name perfectly, so thank you for that. And thank you for the really nice introduction, and thank you all for having me here today. Thank you, Jeanette, for organizing and reaching out, and I'm really excited to share my research with your group. So I will just share my screen and get started. I probably have about 15 minutes worth of slides, and then I'm available after that for discussion as time allows. So let's go ahead and get started. Does everybody see my presentation okay?
Very good, thank you.
All right. So today I'm going to talk a little bit about mass spectrometry metabolomics as a tool for discovering bioactive constituents of botanicals and fungi. And what I'd like to do is actually take a little bit of time to talk about collecting quality metabolomics data sets, which I think remains a challenge for the natural products field and really for the field of metabolomics in general. That's an area that our group has been thinking about a lot lately. And then jump in at the end to talking about integrating those metabolomics data sets with measurements of biological activity to identify active components of mixtures. So to start out, I just need to, of course, acknowledge that the work that I'm talking about here today is not the work that I've done myself, but it's the work of a wonderful team of scientists here at the University of North Carolina, Greensboro, including people from all around the world and undergraduates through postdocs. And I'm very honored to have the opportunity to work with them, and I'll highlight some of their specific contributions throughout my talk today. The other important collaborators we have in this research is fungi and plants that produce really interesting small molecules. And a lot of the work that we do is aimed at trying to figure out how the molecules that plants and fungi make can be used in medicinal contexts. This is just a photograph of fungi growing in that have been grown out from our library at the University of North Carolina, Greensboro. We have thousands of living fungi from all different genus and species, and even different orders, families, and they are making very cool molecules, as you can tell by the photograph that there's different chemistry going on here. And really the central driving force in our work is asking the question, how can we take that chemistry and use it for something beneficial to humanity? And a big part of that work for us is antimicrobial drug discovery. And we also acknowledge that in the natural products field, there has been this discovery void, which probably many of you have heard about if you're at all involved in drug discovery research, in that there was sort of this golden age of drug discovery for antibiotics between the 40s and the 60s, and then things tailed off. And recently there have been very few new antibiotics discovered. And there's a number of possible reasons for that, and there's a lot of discussion as to why the success rate for developing antimicrobials and antibiotics is not as good as it once was. And part of that people argue has to do with the low-hanging fruit having already been picked from the natural product sources. Others look at the investment on the part of private industry in drug discovery and antibiotics, which is not necessarily very lucrative as a money-making scheme for pharmaceutical companies. And there's also sort of been a shift away from traditional approaches of natural products drug discovery towards other approaches for drug discovery, including combinatorial approaches, has not necessarily yielded the results that we would have hoped for. So I think there's sort of a perfect storm of challenges that face this field. And we would love to contribute in academia to addressing some of those challenges. And so a conversation that goes on a lot with me and my colleagues and collaborators and students is this question of whether modern analytical methods, mass spectrometry, and other methods, including genomics, metabolomics, proteomics, whether this will actually revitalize drug discovery for antimicrobials and other areas. And of course, those of us who are in the analytical methodology development field are excited about trying to develop better tools for this. And I guess I leave this as a question to all of you to think about throughout the talk, is how we're doing on this effort to actually make drug discovery more successful based on the modern analytical methods and big data, etc. So I don't think I have to explain to this group what metabolomics is, but I thought I would share my definition nonetheless, because I think it's important that we all know what it is that we're talking about. So when I think about metabolomics, I typically define metabolomic studies as studies where we measure differences in abundance and presence or absence of a holistic profile of small molecules, the metabolites, in a given organism to enable comparison among complex samples. So for me, metabolomics usually means that there's some quantitative component, but it's typically not absolute quantitation. So we use peak area as a proxy for concentration, which means we're usually making relative comparisons in concentration. And then we often don't know what the identities of all the metabolites are that we're looking at. And so we use mass to charge ratio combined with retention time as a proxy for identity when we're looking at individual molecules in the samples. And then maybe we go back and solve structures later if the molecules turn out to be really interesting. So the question that I would pose today is, does mass spectrometry metabolomics really improve the success of natural products drug discovery? Because of course, colleagues, including myself, have been arguing for a long time that it will. So let's think a little bit about how natural products drug discovery happened historically. And really, these are the approaches that were used to discover all of those antibiotics that are listed on the earlier slide as being discovered between the 40s and 60s. What are if that should be the benchmark for what we're trying to improve upon. So how does that work? Antimicrobial drug discovery in natural products was really typically based on doing phenotypic assays from complex extracts of organisms. And here's an example. This is our mycologist, Hussefa Raja, in the mycology laboratory at UNC Greensboro. Here's a fungus, Alternaria dacifolia, that he cultivated. And we identified interesting antimicrobial activity of this fungus in a screen. So the question is, what is responsible for that antimicrobial activity? Well, a typical natural products approach would be to collect a screen of data from that fungus using liquid chromatography, either coupled to mass spec or to a UV detector. and obtain a chromatogram, and then to try to figure out which of these peaks in the chromatogram are responsible for the activity. And of course, one approach would be to go through one by one and isolate each one of these compounds. That could take a really long time. Another approach would be to do what we call bioassay guided fractionation, where you take the extracts, you test them in the bioassay, you get an active extract, such as the Alternaria docifolia extract. Natural products chemists like to call all biological evaluation bioassays. Obviously, there's a lot of variation in what that might look like, but it could be, in the case of antimicrobial screening, trying to kill a microorganism in culture, or in a anti-cancer assay, testing in an animal model for tumors. Then we do some separation of that active extract and generate a series of fractions from it. And then we conduct the biological assay again on the fractions and choose an active fraction from those. And this process goes on iteratively of separating, fractionating, until ultimately, ideally, you have a pure compound that's active and you solve the structure. So the idea of the bioassay guided fractionation is that you're always pursuing the most active fraction and therefore ultimately coming up with an active lead. And that, again, that process is the one that's been used to develop the approved drugs. So although people often criticize it, it does have a track record of success. So how can we do better than that? Well, natural products chemists decide what to isolate in the more typical way of doing it. What they would do is they would generate chromatographic data on a series of fractions, and they would then look at comparing the chromatograms for the fractions to the chromatogram for the extract itself, the original starting material, and look at peaks that appear to be present in the extract, but absent in, and present in the active fractions and absent in the inactive fractions. So again, you're looking at some biological activity data. In this case, we're looking at % inhibition of Staphylococcus aureus growth, and we're comparing the extract, which seemed to inhibit completely the bacteria, and fraction 2, which also seemed to inhibit the bacteria. And this is a simple case where the other fractions have no activity. And so if you just look at this by eye, and I encourage you all to do that for a moment, you could see that, so this yellow trace that represents fraction 2 does have some things in common with the trace for the extract. And one thing that jumps out right away is this peak right here, which is present at relatively high abundance, or at least detected as a strong signal in the mass spectrometer in the extract and also fraction 2. And so we might then go about isolating that compound and seeing if it's active. And that's how natural products chemists would go about proceeding through this bioassay guided fractionation process. So what do the mass spectrometrists do? Well, we make it a little bit more complicated, because the data that we look at is not simply chromatographic data, but we start to look at what's going on underneath all those peaks and what we can detect with a mass spectrometer. And if we look at a chromatogram now and we're using the mass spectrometer as our detector, we can start drawing different thresholds and determining different numbers of ions in that chromatogram, depending on where we draw the threshold. But we can start to get really complex data sets when we look at not just the the chromatographic data, but also the mass spec data. So like if we draw the threshold here at 1 × 10⁹, we only see one feature in the data set. And by feature, what I mean is a unique combination of a mass to charge ratio and a retention time. But if we start to draw lower thresholds for our signal, now we start to see more features. So here we see 10 features above the threshold of 1 × 10⁸. If we zoom in on the baseline, we can see 99 features above 1 × 10⁷. We can see 843 features above 1 × 10⁶. We can see 4,900 features above 1 × 10⁵. And so obviously this is both giving us more information than what we would get out of a chromatogram, but also potentially complicating the data. And a lot of times I would argue that in the metabolomics field, we have data sets that contain thousands of features without really knowing what that means in terms of the actual composition of the sample and which of those features are truly relevant. And so if we're really going to do a good job of using metabolomics data to inform something useful like drug discovery or other applications, we have to have a better sense of what those feature counts really mean and where to draw the line in terms of what's relevant feature and what's not. So why do we see so many features in mass spectrometry data sets? Well, one reason, of course, is that mass spectrometry detects real analytes at low abundance. And you can see here, this is using MZ-Mine to process a mass spectrometry data set and deconvolute some of the peaks that are present below what appears to be the baseline. And what you might see as the baseline in a UV trace would be somewhere around this level. And you see there's definitely some real peaks down there that could potentially be relevant, or they might be too low abundance to be relevant. But of course, I say low abundance here, but we also know that mass spectrometry can be deceiving, and abundance in the sample is not necessarily correlating to the abundance of ions that we detect. So it's also possible that some of these features down in the baseline are actually present at relatively high abundance in the sample, but are only giving us low signal in the mass spec. Of course, there's also this issue in mass spectrometry of redundancy. We see multiple features per analyte. And thirdly, we detect contaminants that aren't relevant analytes in our samples. And so one of the things that we've been thinking about in terms of doing a good job of mass spectrometry metabolomics is really to address those questions and figure out ways to come up with good feature lists in metabolomics, and to determine how to remove the interfering ions, how to group together the ions that are associated with a single analyte, and ultimately to get quality metabolomics data sets. And this is actually an ongoing effort from my research group, but that's been part of the Center for High Content Functional Annotation of Natural Products. In recent times, we are funded by the National Institutes of Health with this center, collaborating with Roger Linnington and John McMillan. Roger Linnington is at Simon Fraser University. John McMillan is at the University of California, Santa Cruz. And the project that I lead as part of this larger grant is all about thinking about how to get quality data out of metabolomics. So I want to talk a little bit about a project that we've been doing along these lines. And this is the test case project for some of the examples I want to show today. So what we have actually as our data set is 38 sources of green tea. So imagine this as tea bags that you might find in a grocery store if you were going to buy green tea. Also supplements, which are pills that contain green tea extract that people consume for various reasons, assuming that they somehow have a health benefit, antioxidant health benefits or other health benefits. And we purchased all of these samples as part of a project actually to do a clinical study on the impacts of green tea on metabolism of pharmaceutical drugs. And that study has been done as part of another project that I'm not going to talk about today, but we're also using that same data set as the basis for looking at complex metabolomics data sets. So obviously there's some similarity in the green tea samples, but also some differences depending on the source and the way that the samples have been prepared. So to evaluate that, we made triplicate extracts of all the tea samples from different sources, and we extracted all of them in methanol. And we took a set of the samples and we sent them to our collaborators at Simon Fraser University. We kept a set of extracts at UNC Greensboro, and we did some data acquisition with mass spectrometry, data processing, and data analysis, and compared across the laboratories. And we've actually recently published the results of this in the Journal of Natural Products. I'll talk a little bit more about that in a bit. So I want to first of all start with this data set and taking a little bit of a deep dive into the data from one laboratory and asking some of these sort of what seem like simple questions, but are not necessarily completely agreed upon within the field. One of them is how many features do we really see per analyte in these data sets? And the second is how can we remove contaminant signals and improve data quality? And Joël Hourier, who's a postdoc who was working in my research group and is actually now still with us remotely from Switzerland, has done a lot of the work here. This is her working during the pandemic in our community garden, where we spent a lot of time collaborating because we couldn't be together inside. So first question, how many features per analyte? So again, here's an example of chromatography data where we've actually, this is with the green tea project, a simple example where we have a mixture of standards that are all components of green tea. And those are all combined at an equimolar concentration of 100 micromolar. We analyze them 3 times, and these are just 3 chromatograms overlaid on top of each other. And here's showing the data in the way we typically look at chromatographic data, where we have time and then intensity in the mass spectrometer on the y-axis. But we can also look at these in a 2-dimensional data plot. And in this case, we can put the retention time on the x-axis and the mass to charge ratio on the y-axis. And now you start to see that there are multiple different features, even at a given retention time. And the way I'm going to be showing data for this project moving forward here is looking at what we have now here is a 2D feature map, where each one of the features is shown as a dot, where we have intensity on one axis and time on the other. And the size of those dots is indicative of the abundance of the feature. So something like caffeine and theanine, which are very high abundance components of the green tea sample, give us big dots. So here's just an example of what the 2D feature map looks like for a sample where we injected just caffeine, one molecule. So, you know, people talk about having these really complex metabolomics data sets. This is using an Orbitrap mass spectrometer, a Q-exactive, and acquiring a data set on a single analyte. And we see 805 features for just one analyte. So I think that's right off the bat, a little bit of a wake-up call to what it means when we see large numbers of features in a metabolomics data set. Now you can see that not all of these are associated with caffeine. They're spread across the chromatogram. We see a lot of features in the front. We see a lot of things coming out later. Everything in this region is associated with caffeine. And you can see this is the actual mass spectrum for caffeine, and only the features in orange were annotatable. So we only were really able to annotate about 3 of these features, but we assume that others are also associated with caffeine. But this is a pretty simple example. When we look at another analyte here, gallic acid, so this is injecting again a single analyte of gallic acid. We see an even higher number of features for this sample. And you can see in orange all the features that we could annotate amongst those, which is a lot. In fact, if you look here, this is just the mass spectrum for gallic acid, and all of these ions are given an annotation. Some of them are rather unusual annotations, but they seem to match based on accurate mass, these annotations. And if we zoom in on the x-axis, we can keep zooming in and detecting all of these features that appear to be associated with gallic acid. And we stopped after annotating 100 features associated with just this single analyte. This is all in MS1. We're not doing any fragmentation here. So it's interesting, gallic acid is not easily ionized in the positive ion mode. Caffeine is. We see a much more complex data set for gallic acid. And I think what that is saying is that probably, I hypothesize anyways, that when analytes are difficult to ionize in an easy way, we end up seeing a lot of strange addicts in the mass spec, and we can have a lot of redundancy in the features, over 100 per single analyte. So there's that redundancy issue. And then there's the question of whether all the features are actually real. So there's a possibility that we have contaminants that are coming from the air around the mass spectrometer, from the liquid chromatography separation. Of course, there's also a possibility of contaminants being present in the sample themselves. Usually the way that we think about removing contaminants is to simply filter the sample to remove anything that's present in the blank and not present in the sample above a particular threshold. But some of the work that we've been doing recently suggests that that's not enough when it comes to cleaning up metabolomics data sets. And I just want to show you some examples of that. So here's an alternative approach that we've been using, which is a quantitative filter, requiring not just that the feature of interest be present in process replicates in 3 separate analyses of the same, of replicate extracts of the same sample, but that it be present with a relative standard deviation value above a particular threshold. And so we've been doing data processing where we keep features that we analyze process replicates of each sample. And by process replicate, what I mean is a replicate all the way from extraction through to analysis on the mass spec. Analyze 3 process replicates of each sample, and we look at how well the RSDs cluster on those 3 replicates, and we reject samples that don't cluster well, and we reject features that don't cluster well in terms of the relative standard deviations, and we keep the ones where the relative standard deviation is below a certain threshold. So here, this is an example where the relative standard deviation is poor across the 3 peaks, and here's where it's good. So this one would be kept, this one would be rejected. And we've shown actually, and I'm not going to get into the specifics of this today, that a simple qualitative filter where you just look for the presence in all 3 samples is not really sufficient for cleaning data sets, but that it's really important to look at the peak areas as well. can see that here. This feature is there in all the samples, but the abundance is really differing from one sample to the next. So here's an example of using that quantitative filtering approach with relative standard deviation using a reference compound mixture where this is the example I was showing earlier where we have all these different analytes that are known to be in green tea. We've made a mixture of them at 100 micromolar and we've injected it into the mass spectrometer. And I'm showing a 2D feature map here where each feature is represented with the size of the dot being indicative of the abundance. And what you can see, if we look through this simply, if you look at everything in the sample that was present in the blanks with relative standard deviation of less than 30%, meaning that it's a real peak in the blank, we remove everything that's found both in the blank and the standard, and that's shown in the orange dots here. If you remove the things that are detected in the blanks only, that's these. If you remove, we also look not just at the presence in the blanks, but at the ratio of what's present in the blanks versus the standards, because there are cases where you have carryover and you can have an analyte that was present in the blanks simply because there was a little bit of carryover from the sample and you don't want to remove it because those are often the important high abundance samples. So if we filter the sample in that way, what we end up with is this is what the original data set looks like. These are the ones that we remove. And here's the filtered data set. Now let me show you a more complex example where we're not just analyzing standards, but we're actually looking at a true extract of green tea. So this is a much more complex sample set where we've got a real biological sample, an extract from green tea. And again, showing all of the features in here that would be removed, but based on this filtering, based on relative standard deviation. This is, again, the sort of illustration that it really matters where you set your threshold. Here's a feature list where we set our threshold at 5 × 10⁵. That's the mass spec response. Of course, it's instrument dependent, so these are just arbitrary values, but it's a sort of a middle height value for our instrument. And here's a low height value, 1 × 10⁴. And you see quickly that there's a lot of really poor quality features shown with the red dots that start showing up when you set a low threshold in your signal. And if you compare that here to what happens just after blank filtering, what you see is that filtering based on the blank did not remove all these crummy features at all. They're still there, even though we use a blank filter. And here's what data sets look like comparing top and bottom, where you've actually used relative standard deviation of the peak areas as a quantitative filtering approach. And you can see how much simpler these data sets are once they've been filtered to remove the interferences from the data set. So these are data sets now that have only features that had a relative standard deviation less than 30% in triplicate process replicates. Why does that matter? Well, if we want to look, for example, at a feature-based molecular network for the green tea data, so this is generated using GNPS, which is obviously a common processing method used on metabolomics data sets. And the point that I want to make here is that I think there's a lot of GNPS networks that people create on the data sets where the rigor of the data is not very good, and so you can start to draw incorrect conclusions. For example, so this is the feature-based molecular network of the green tea data. Here's this is without cleaning up the data sets, what the network looks like. And this is just showing, highlighting in yellow, all the features that would have been removed based on blank subtraction and all the features that would have been removed based on the relative standard deviation filters. So quite a bit of the data set. Here's an example of creating that same network now with a cleaned up data set. And actually here's generating the network with the cleaned up data set. And this is just the network where we've actually physically subtracted all of the blanks and the features that were in the blanks and removed by the filtering. And what you see right here is that the connectivity is totally different when we do molecular networking on a data set that was cleaned up first. And a specific example of that is here. So this is just a single cluster within the network where you can see that a bunch of individual the yellow boxes are features that were present in the blank that are showing up as part of this network, even though in theory they shouldn't, because they shouldn't have MS2 data matching actual analytes. And here's a case where some of the features in the red boxes are poor relative standard deviation across replicates features. And here's what happens when we actually have looking at those same clusters where we've actually filtered the data first prior to generating the clusters. And what you see is that now the connectivity is different in the data sets. So I guess this again makes the point that you can be coming to erroneous conclusions about what's going on with your sample if you haven't collected it in a rigorous fashion. So the take-home message from that first part is that the data pre-processing and the removing of the unreliable features can really improve the interpretation of mass spectrometry metabolomics, and that it's not enough to just subtract what's in the blank. So now getting into this question of comparing data sets across laboratories, which is another ongoing effort as part of this HiFan project that we have from the National Institutes of Health. Again, just to remind you, this is going back to the major data set. We have all these 38 sources of green tea. We've sent extracts to UNC Greensboro and to Simon Fraser University. We have an Orbitrap mass spec platform at UNC Greensboro. Simon Fraser uses a QTOF. We both have Waters Acuity front ends on those systems. And we recently published this paper about our findings for comparing the data sets that we generated on those green tea samples across labs. And I just thought I'd show you a couple slides about that. If you want to learn more about it, you can check out the paper. So one of the things we had to figure out in this data set was how to compare features across the laboratories, because we see very different, and I'll show you this in a minute, we see very different sets of features per analyte, even in the MS1 data, when we collected in different laboratories on different platforms. And so what we determined was that we were able to correlate retention times very effectively across the two laboratories because we were using the same column and the same chromatographic method. And so we actually decided which features were correlating with each other based on retention time matching as well as mass to charge ratio. And that was Trevor Clark in the Lennington lab who did that work. And I just want to show you here a snapshot of how the mass spec data compared across two labs. These are, again, data with the standard mixture of standards that I showed you earlier. So these are all different standards that are present in green tea. And what I'm showing is these similar kinds of plots to what I was showing earlier, where we've got mass to charge ratio and retention time here. And what you see is A and B are laboratories A and laboratory B. And the blue dots are, so laboratory A is the lab at Simon Fraser University. The blue dots are features that were present only in the Simon Fraser data for that analyte. And the green dots are features that were only present in the UNCG data. And then the orange dots are ones that were present in both. And what you see is that the features that we obtain in the MS1 data for these analytes are dramatically different from one platform to the next, perhaps not too surprisingly. And that makes comparison of feature lists across laboratories really challenging. And we think that that's because of different ionization efficiency and cluster formation and fragmentation that's happening in source on the different mass spectrometers. But interestingly enough, if we plot principal component analysis representations of the data sets from the two laboratories, so now here what we're showing is each one of these dots represents a replicate of the sample. So shown in principal component analysis, basically many of you have probably seen these projections before, but the closeness of the dots to each other indicate the chemical similarity of the analyses based on the data that's fed in, which is the mass to charge ratio and the retention time profile for each sample. So what we see is this is the data from UNC Greensboro, this is the data from Simon Fraser, and we have a bunch of samples from tea leaves that are all clustered similarly in a similar portion of the PCA plot. We have a bunch of samples that are green tea supplements, so these are capsules. They're clustering together, and then we had a sample that was not green tea, and the triplicate analyses for these are all clustering separately outside of the 95% confidence interval circle. So essentially, the conclusion I draw from that is that although our feature lists are very different from one lab to the next, we can make qualitative comparisons across the samples within a lab very effectively nonetheless. So essentially we can still draw conclusions about similarity and dissimilarity of samples using these very different feature lists, but we should be very careful about assuming that we can compare feature lists across laboratories because we're going to see dramatic differences depending on how they're ionized within the source. All right, so now the last part of my talk, what I want to do is move away from this sort of best practices of metabolomics piece that our lab has been thinking about and into this question of, which was sort of the central driving force for the talk, this question of correlating the chemistry with another dependent variable, for example, biological activity, with the goal of actually identifying active constituents of a mixture. And I bothered to take the time to share the first part, partly just because I'm very curious whether sort of the findings we have are mapping on to the experience that you have in your research groups. I know that you're very much leading experts in metabolomics data collection, and also because our experience, and I'll show this in a minute, is that if you don't do a careful job of the data acquisition part on the metabolomics up front, then attempting to start correlating metabolomics profile with biological activity is very, very challenging and not very fruitful. So we have a collaborator, Dr. Olav Kvalheim at the University of Bergen in Norway, who's an expert in multivariate statistics and informatics. And we've been working with him for a number of years on thinking about the best algorithms for correlating metabolite profile with biological activity. So I want to share a little bit about the outcome of those studies. And I want to go back to the original example that I shared at the beginning, this question of what's responsible for the antimicrobial activity of this fungus, Alternaria docifolia. So we talked about how natural products chemists might approach this. They would fractionate and pursue the bioactive fractions and try to isolate pure compounds. What if we try to use metabolomics as a tool to do this instead? Well, we can generate a matrix of data on all of the extracts and all of their fractions. And that matrix looks something like this. And I'm sure many of you have handled matrices of this kind in your past. And maybe I'm giving you some, flashbacks, post-traumatic flashbacks as you look at these data. But you have mass to charge ratios of many different ions that can be detected in the sample. You have chromatographic peak area for each one of those ions. And then in addition to that, we have some measured biological activity, which in our case is essentially going from 0 to 1 of the extract and the fractions. And if you'll recall earlier, fraction 2 was very active, so it has a high level of activity. Activity of 1, that's just the ratio, the activity of the vehicle, the sample, and then the extract also has high activity, and then these fractions have very low activity. So to integrate these two data sets together, what we end up doing is using this selectivity ratio, which is an approach that Dr. Kvalheim developed and published some years ago. And effectively what we do is we do partial least squares analysis of the data, and then we plot the selectivity ratio, which is the ratio of the variance that's explained by the model to the variance that's unaccounted for by the model. And the qualitative meaning of that is that the selectivity ratio is a measure of the extent to which a detected ion is correlated with biological activity. So a high selectivity ratio means a good correlation between the biological activity and that particular feature, and a low selectivity ratio means that the feature is probably not very important in the biological activity. Of course, that's all correlative, so it has to be validated, any predictions that are made based on that. So here's an example of our LCMS chromatogram of the Alternaria docifolia extract. I showed you this chromatogram earlier, but here's when we do the selectivity ratio analysis on the data, we can plot this chromatogram now using the selectivity ratio as the y-axis instead of the abundance as the y-axis. And what you see is that it looks very different. No longer does this peak here seem to be the most important peak. Suddenly we see some peaks here that are showing up as being very interesting. They are associated with features with masses of 400.28 and 422.22. That might seem like a no recognizable mass difference to some of you who are listening in. So peak number 1 is no longer seeming to be the important one. Now we've got this other peak here that seems like it might be important based on biological activity. And another way to represent this data is more like a mass spectrum, where instead of plotting the selectivity ratio as I did here, selectivity ratio versus retention time of the features, we can plot it as selectivity ratio versus mass to charge ratio of the features. And again, these same two features come out as the highest selectivity ratios, and that one of them is an ion here with mass M, and this is the sodiated version of that ion. So we actually went and validated this approach of using the selectivity ratio to predict active compounds in the Alternaria docifolia mixture. We isolated peak number 1, which turned out to be Alternaria monomethyl ether. And you can see here we tested the antimicrobial activity of that molecule alone. And this is the OD600, this is concentration. So basically the more inhibition of OD600 you see, the stronger antimicrobial activity the compound has, because this is a measure of the bacterial growth based on turbidity of a solution. And we compare that to this feature here that was predicted as being active based on the informatics analysis of the data. And you can see that that is actually altercetin, which is a much more potent antimicrobial than Alternaria monomethyl ether. So if you had just gone through and tried to isolate all these peaks one by one, you probably wouldn't have gotten to altercetin very quickly, because it doesn't necessarily pop out as one of the more important peaks, but we could actually predict that it had a strong correlation with the biological activity, isolate it, and it turns out to be active. So then just taking that to another example, this is a project that was done recently by one of the former graduates in my research group, Manid Kin, who is now a PhD student at the University of Illinois, Chicago. And Manid was looking at Rosmarinus officinalis, which is rosemary, which has antimicrobial activity. In fact, you've probably all consumed many products that contain rosemary because it's used as a common preservative, a natural preservative to prevent microbial growth in food products. And we were interested in applying the same approach I was showing with the fungal extract to identifying antimicrobial constituents in Rosmarinus officinalis. And what I want to demonstrate here is this is one of these selectivity ratio plots that I showed you earlier, where we're showing the selectivity ratio as a function of mass to charge ratio. And this is without prior cleaning of the mass spec data, just taking the mass spec data without removing the features that have poor agreement in relative standard deviation. And we get a pretty complex representation here that's hard to interpret. And this is truly where we were stuck on this project at first when we looked at the data set. But then we tried using this cleaning approach where we remove all the features that have poor agreement in their relative standard deviations. And you can see how much more simplified the selectivity ratio plot is. We've got some redundancy here in terms of features that are associated with the same analyte. We've got analytes 2, 3, 4, and 5, and those all were actually isolated and the structures were solved. And some of them are known antimicrobial compounds from rosemary, and others are newly discovered antimicrobial compounds. So the main point here is that the cleaning of the data sets really helped us to be able to make useful conclusions when we're trying to integrate biological and chemical data sets. So we can use the selectivity ratio analysis, but parts 1 and 2 of the talk are not unrelated, because if you don't do a good job collecting your metabolomics data in the first place, you will have a garbage in, garbage out scenario. So I sort of end here with going back to the original question I posed, which is, does mass spectrometry metabolomics really improve the success of natural products drug discovery? I don't think we necessarily have an answer to that yet. I think the field is still struggling to address some of these challenges, like producing quality data sets and doing meaningful interpretation of the resulting data. But I think we are getting there. I also think that the data sets become so much more complex when you look at them from using a mass spectrometer as your detector instead of, say, the old school UV detector or TLC, that you can end up with a scenario where you get let off on sort of chasing wild herrings that don't end up actually yielding anything. And so we have to be very cautious of that as a field. Again, I want to thank my research group one more time, and also acknowledge the funding that we've had to do this work, primarily from the National Institutes of Health. And now I will open up to any questions that you all may have.
Thank you so much. That was really tour de force and very informative. Traditionally here, Dr. Arpana Vania, our outreach coordinator, would steer the discussion. So Arpana.
Yes, thank you, Oliver. Great talk, Dr. Cech. I really enjoyed it, and especially your approach on starting the talk with the best practices that you use in your lab. I think it's very important to kind of set that foundation in terms of, you know, before prefacing talking about your data and what you've done in your research. We have a couple of questions in the chat. There was one in terms of referring to your slide on the bioassay guided fractionation workflow. And Jenny had a question in terms of how many cycles of bioassay fractionations are usually required for one botanical sample?
Yeah, great question. So typically what would be done with a botanical sample to try to isolate a pure compound with bioassay guided fractionation would be to extract with methanol, then do liquid-liquid partitioning in about 3 different stages, removing the lipids, then removing the tannins, and eventually ending up with a somewhat purified organic fraction. And then that would be separated with flash chromatography using a silica gel, so a normal phase separation, 1 or 2 stages of that, followed by reverse phase preparative HPLC, often 1 or 2 phases of that. So, you know, there's the liquid-liquid partitioning step, and then usually 3 or 4 stages of rounds of chromatography and bioassay before you actually end up with a pure active compound, if you're lucky.
Yeah, so it seems like a lot of steps. And then to kind of follow up with that question, ideally, what is the minimum and maximum number of fractions for a given botanical sample to capture as many features as possible?
Usually it's around a dozen or so, 6 to a dozen fractions per step. I would say, so to your comment about it being a lot of steps, it is a lot of steps, but I think that, you know, we've often talked about bioassay guided fractionation as being very laborious, but at the same time, you know, creating these giant metabolomics data sets and trying to interpret them is laborious too. It's just sort of a different type of labor. So
Right.
I in my experience, it's funny because I'm the mass spectrometrist, right? So I believe in metabolomics, and I've been trying throughout my career to use it in effective ways to aid in drug discovery. I have a colleague down the hall who was trained by Dr. Wall and Dr. Wani, who discovered 2 of our most important cancer drugs, Taxol and camptothecin, and he does it the old school way, and he outcompetes me in terms of how many pure compounds he discovers and solves structures of by orders of magnitude. So I guess that's my personal perspective, is that we often say, oh, this bioassay guided fractionation is so laborious, but the fact of the matter is, it's the labor to accomplish something that works. And I would love to say that metabolomics has made it less laborious, but I don't think it necessarily always does.
Yeah, I mean, it ends up being laborious towards like the data analysis. Yeah, right. Like the number of features you were talking about and how to reduce that.
Yep.
So yeah, there was a question in terms of when you were talking about how you remove contaminants in your data set, specifically the RSD filtering. Are those replicates injected directly one after the other, or could some of the variations be explained by sample degradation over time?
Oh, they're all analyzed in the same mass spec run. And so you'll see some features that are very consistent and others that are inconsistent. I think it's a great question though, where are those inconsistent features coming from? And what we generally find is that they are contaminants within the system of some sort. So they come from the LC column or from the solvents or from the atmosphere, and we can actually identify them as being on the list of common contaminants often when we dig deeply into what they are in mass spec. They're often polymers and things like that. So I don't think they're so much due to degradation. I think that what happens is if it's not actually a component of the sample, then it doesn't get separated and identified consistently from one injection to the next. And that's how you can find it, is based on the fact that peak area is not consistent.
Great. There was a question on PLSR and how it looked to explain linear covariance between bioactivity and the feature areas. Is this correct? If so, to what extent do you think assuming linearity is a fair assumption?
Maybe the person who asked that question could repeat it. I'm not sure I completely follow it. Oh,
Yeah, Parker, if you can unmute yourself.
Yeah, so basically you had those bioactivity like Y variables, and then you had the like peak area X variables, right? And you're doing partial least squared regression, if I'm not mistaken.
Yep.
And so my thought is basically you showed like what I think is basically the R squared value, right? Which is like the explained covariance related to the unexplained covariance. And so I was just thinking, it's a linear model, you know, to what extent do you think assuming linearity is a fair assumption, essentially?
Oh, yes. Now I follow
Or
you
you consider
now.
trying something nonlinear?
I follow you now. So really excellent point. And I think that there are cases where the relationship between response and feature area, which is a measure of abundance, may not be linear. And certainly if we have saturation in the signal in the mass spec, for example, or if we have ion suppression happening, those could impact on the linearity. There's probably other examples we could think of. So that's a great question in terms of trying other models. And I'm not sure whether we really have done that. So that's definitely something to think about down the road. But it's a really good point that that's an inherent assumption of this model.
I have one quick follow-up if I could.
Yeah, go ahead.
Essentially, I wasn't actually thinking about in terms of the nonlinearity of like the feature creation. I was just curious, like, does biology work like that? Like, is it like linear in that sense?
That's a fair point too. Is that, do you actually get improved biological activity as you increase concentration? And I think, you know, it's a classic chemist statement to ask the question if biology behaves like that, because of course the answer is sometimes it does and sometimes it doesn't. And that's the question I would ask. That's the way I would ask it too. I'm classifying myself as a classic chemist. So, you know, I think that we know dose response curves are sometimes linear and sometimes not. There may be better models based on the dose response relationships as well. Yeah.
Curious, thanks.
Parker. Yeah, excellent point.
Great, thanks, Parker. There was one question in terms of thinking about the complexity of plant metabolome, how it's so much larger than the animal metabolome. So when you're filtering down your feature list, maybe down to 300 metabolites, does this hamper or kind of affect your understanding of the metabolic features that you're finding in plants? And then will increasing the number of fractions help you kind of retain more information later down in your analysis?
Yeah, those are great questions. So I think definitely that having a larger number of fractions does help us to detect more features that are actual components of the sample, because of course you're boosting the abundance of everything in that fraction and improving the ability to detect the likelihood that it would be present above the limit of detection. So yes, having more fractions could certainly help, and we've done that. The downside of having more fractions is, of course, if you're doing things based on biological activity, you have to have sufficient material to bioassay each fraction. And so that actually becomes the practical limitation, is conducting the biological assays and having sufficient material to do that. So if you have an assay that's tolerant of small amounts of material, you can make more fractions, then yes, that of course helps. In terms of, you know, whether we're actually capturing the complexity of the plant metabolome, that's a question that, you know, I still have in my mind for sure. And I don't really know, I would be curious for what others who are on this call think about how many secondary metabolites we should be detecting in a plant extract. Obviously, it depends on where you set your threshold as what counts as a real analyte. And I was showing in the data sets earlier that depending on where we set our threshold, we see more or less features. And of course, at the lower levels, we also have more features that aren't real. But it's an ongoing question for me. What is a reasonable number of analytes per sample? And I think because the field of mass spec metabolomics has usually looked at samples as a feature list rather than an analyte list, I'm not sure we have a really good answer to that question yet.
To
But it's
piggyback
something we
off,
should continue to think about. Yeah, go ahead.
To piggyback off of the threshold comment, do you normally in your practice change your threshold depending on the type of sample you're analyzing, or do you have a routine threshold that everyone uses when you're processing data?
Great question. Yeah, so we're actually working on a paper right now where we're talking about the approaches that we use for setting the threshold, because I think that's such an important question, and everyone who does metabolomics has probably sat there in front of their data set and said, okay, where do I set this threshold? And if I set it too high, then I miss things. If I set it too low, it's going to take 10 years to process this data set, you know. So one thing I think that we have found, I can just say this qualitatively, is that if we set a relatively low threshold, but then do a really good job of cleaning up the data to remove the features that are not consistent across replicates, that seems to give us a better data set than if we were to just set a higher threshold to start out with. So in other words, there are real components of the analyte that are present at low levels that we think are still important that we can detect when we set low thresholds. But the question, which is a good one, was, you know, do we do that in a data sample by sample measurement, you know, empirically decide, or is there some rule for where to set it? And I think we usually decide what the best parameters are for each separate analysis, depending on the question that we're trying to answer and the quality of the data set. And so I personally don't think that there's a threshold out there that we can say we should always use. And of course, every mass spectrometer, you know, these 10 to the 5th or 10 to the 10th or 10 to the 8th signal values are completely instrument dependent anyways. And it's not clear exactly how they correspond to the numbers of ions. So yeah, I think that we have to decide with each data set what's the right threshold for the question we're answering.
Yeah, I definitely agree with you that, you know, kind of setting it low and then cleaning up your data is probably the best, and tailoring it per sample type. There was a question regarding the fungi that you had mentioned earlier in your talk. Generally, how do you identify fungi of interest in your lab? Is it similar to what Dr. Gary Strobel does with his work on medicinal endophytes?
Great question. Yeah, we do have a number of endophytes in our library. And of course, I find Dr. Strobel's work to be very inspiring. We actually have done a lot of work on a fungus Dr. Strobel did the original characterization on. Our fungi are mostly aquatic fungi. They've been collected from rotting wood in both freshwater and saltwater sources. And part of the reason we've done that is because we've discovered that we get some very different fungi from what are in other people's libraries. So also we do have a number of endophytes in our library, but we sometimes find that the endophytes are not very consistent in terms of their ability to continue producing secondary metabolites over time. And we think that's because, you know, they have these specialized relationships with their host plants. And so sometimes they don't behave as well in a culture setting, but that's not 100% true across all of biology. Of course, it depends on the organism. So yeah, we sort of have taken a random approach to acquiring as many fungi as we can rather than focusing on a single family of fungi. And then we try to prioritize the ones that we look at based on them having interesting biological activity.
Great, thank you. A question just popped up in the chat referring to what extent of phylogenetic relationships or genomics or systems biology can help guide discovery of natural products?
Yeah, I think it's a great question and a great point. You know, we have been very much metabolite driven and biological activity driven as opposed to looking at the genomics and system biology in our efforts. But I think, you know, I guess I would just turn that question back around to the people who are working in those areas. I find it fascinating. I also don't know how much that has played out in terms of actual drug discovery for small molecules yet. But I think that there are still lots of potential there. And certainly when we look at what's going on with access to the coronavirus vaccine, we can appreciate the importance of, you know, the genomics revolution. So yeah, no, I don't think I have an answer to that, but I think it's a question we should all be continuing to ask ourselves.
Yeah, I mean, it's definitely plays into our understanding of metabolomics and in general, so it's very important to think about those things. There was a question, there was a question from Oliver in terms of how you assign addicts to MS1 ions. There was definitely some interesting slides that you showed. Yeah. Where there was like some really crazy addicts.
Bizarre addicts. Yeah. So it's a great question, Dr. Fien. So I have a postdoc who did all that annotation work. It was very involved. She has a couple of papers which I could pass on to you that she used to do that work. Essentially what we're looking at is just a matching in mass between a possible adduct and the accurate mass that we measure. So it's not an absolute confirmatory assignment. It's just that the accurate mass matches. I think it's not an area that is very fully developed in the mass spec field. You know, we have these software approaches that will allow us to assign the most common adducts, but we miss, I think, a lot of adducts and fragments with those approaches. So my feeling is I thought that the annotation part was going to be easy in these studies. I did not anticipate finding more than 100 adducts and clusters and fragments associated with a single feature in the MS1 data. That really surprised me. And I think that one could say, well, maybe those aren't really real. Maybe it's just those are figments of the data that are just fluctuations in the baseline or something like that. But, you know, what really is convincing to me is the difference between caffeine, where we see just a few annotatable adducts, and gallic acid, where we see hundreds. That suggests to me that the annotation process is not just coming up with random hits, but there actually are real adducts that we're seeing. So I think it's MS Cleaner that Joël Hourier used, and there's a paper on that, which I'll send your way after.
Great. So like to kind of continue this conversation on adducts, when you are finalizing your data set, do you combine those adducts? I mean, for gallic acid alone, there was like hundreds of, you know, adduct types. I think one of your slides just showed N+H when you were showing the reduction plots. So
yeah,
what are you doing
great question. Great question. So we typically will look at, so the nice thing about the selectivity ratio plots is that it simplifies our list of features dramatically to look at those that are actually associated with biological activity. So often though, what you'll see, and you probably saw this, if we go back, let's see, hold on, let me go back to a slide here. Yeah, here's a good example. So you can see in here, we haven't removed all of the clusters and adducts from the data set in order to generate these selectivity ratio plots. And you see that where you see, for example, for compound 1, you see both the N+H and the N+H minus water. For this compound, we're seeing the sodium and the ammonium adduct. And you might say, well, shoot, if there's 100 different adducts associated with a feature, how come you don't see 100? Instead, only 3 here. But I think these data sets are that what we're finding is really the major features per analyte that are associated with the biological activity are the ones that are popping up with the selectivity ratio analysis. But I personally like this redundancy. It's a nice gut check to see that we're not just finding a single isotope, let's say, or a single feature, but multiple redundant features, because if the analyte is associated with the activity, then all of the features that are associated with that analyte should be associated with the activity, if you know what I'm saying. So personally, I like that, but I think there's also value in data sets where you could simplify them down to look at them as a per analyte basis instead of as a per feature basis. And that would certainly help when we talk about comparing data sets across laboratories, because the analyte list should be comparable even if the feature lists are not comparable. And that's actually something that we're looking at pretty extensively right now as part of this HiFan project. And we have a software approach in development for doing that. There's some other approaches out there like Camera and RamClust that try to do that. Our experience is that they don't fully address the problem of associating all the features that are of grouping all the features that are associated with a single analyte. So I don't know what your experiences are, though. Maybe you have a solution to that problem. We'd be curious to hear if so.
I mean, in terms of grouping addicts, right? We usually use MSFlow as kind of like our resource in terms of cleaning up data, post data processing, which we use MSDial for that in terms of deconvolution and all that. So MSFlow will flag addicts that, you know, as a user, then you can go back and look and check if they should be combined or removed. So it does provide a lot of flags, but still a little bit of a manual process there. Yeah. And we have resources. There's a paper as well that I could send you in terms of how to use MSFlow. Yeah, so that's kind of our workflow. There was a question, more of like a teaching moment, I guess, here. What is the difference between Pca and Plsda? So there was a question from one of the attendees who wanted a little bit.
I'll do my best on that. So as a mass spectrometrist, my mass spectrometrist 10,000 foot view on that is that principal component analysis is effectively a way of looking at the similarity of or differences of the individual samples within your data set based on their chemical profiles. Well, it wouldn't have to be a chemical profile. People use principal component analysis based on any set of descriptors. You can compare Bible verses with principal component analysis based on the commonness of words that show up in them. But what we do is we look at the masses and retention times that are in a sample as descriptors of the sample, and then we do principal component analysis to simplify, essentially go from a large number of possibly correlated variables to a smaller number of uncorrelated variables that describe the data set. So it's a way of data reduction. Instead of having to describe the data with a whole bunch of features, we describe it with fewer number of composite principal components that describe it. So the dots in a PCA plot represent how similar or different the samples are to each other. And you can think of each one of those dots like a single chromatogram. And if the chromatogram is really similar, the dots are close together. The chromatogram is really different, the dots are far apart. When we're doing partial least squares analysis, now we're actually incorporating the biological activity into that. So we're looking at an independent and a dependent variable. And what we're looking at now in the data set is how well the different features are correlated with the biological activity. And we find that the features that have similar biological activity, or the samples that have similar biological activity, cluster together. And so the partial least squares, you can sort of think of it like, when you think of least squares analysis, you think of linear regression. So you have an X and a Y, concentration and response or something like that. And you're looking at the relationship between those two. But this is just a multivariate way of doing that. So rather than a least squares analysis where we have one X and one Y, we have many, many X's, and we're looking at how they're all contributing to the overall Y. I don't know if a statistician would have at all liked the answer I just tried to give you, but it was my best shot.
We might have a couple on the call. They might be able to.
They might, yeah. You might want to talk to them, Hussein.
They might be able to.
They're probably like, oh.
Yeah. So a lot more detail. So I think you were. It was fine in terms of.
Okay, good.
Explaining the differences. I had a last question before we kind of wrap up here. In terms of your now changing your feature list to an analyte list, what libraries did you use? I'm biased because I've made a lot of libraries in my time as a researcher, and a lot of them are focused on natural products. So I'm just kind of curious in terms of what libraries are you using for identification of your MSMS data?
Oh, absolutely. Yeah, so we try to look at as many libraries as we can to find fragmentation patterns that will match our analytes. Let's see, we obviously search against GNPS. We also use the Dictionary of Natural Products to try to compare our accurate masses against known possible accurate masses. That doesn't get you to the point of MSMS data, but it will give you putative structures based on accurate mass. I think I would actually probably have to turn that question around to the group in terms of other databases that they search, because I know we have sort of multiple ones that we look at. But do you have some that you suggest as being the best?
Yeah, so a lot of the ones that I've created or the ones that we create in the Fiend Lab are all available on MONA, which is Mass Bank of North America. It's a freely available database that has many libraries, different collections of libraries that are, some that are very specific to natural products. The one that I've created has the name VFNPL, but we can definitely talk about that offline. Yeah,
I would love to. I know we've done some with Mass Bank in the past and also with posting data in Mass Bank, but I'm not familiar with BFNPL specifically. So
yeah. So that library, right? So that library that I created was about 3,000 natural product standards over Q-Exactive and QTOF data. We also have MZRT information and a library that we use in-house that's available on MONA. But we use a PFP column for that. But yeah, love to share those resources and kind of chat about what databases you're using.
Awesome. That's super helpful. Thank you.
Wonderful. Yeah, of course. In the last couple of minutes, Oliver, if there are any questions from you directly, or if there's any more
I wish I would now be able to continue the discussion with Nadja for another hour or so, and then show around and have a real tour and maybe a dinner, the classic way how we like to host our guests. Unfortunately, we have to postpone this, Nadja. Maybe when times get better, you come to California and we'll I
would love that.
Do that then. Yeah, so I think we have discussed quite a bit.
Yeah, it was a great discussion.
Like fundamentals of mass spec, electrospray, Addicts, cleaning. And it reminds me, I have tomorrow a 1-hour discussion with the NIH about why we clean so much and why it takes so long time to look at the data, and then another meeting with the NIH on Friday to discuss what we think about confidence and what it means and why we sometimes change our opinion. And why don't we use simply some automated tools like what the proteomics people do or the genomics people do, and why do we make such a fuss? And it is not a short answer other than it's complicated. And yeah, we as a laboratory, Parker, you, other people in my lab, we think we really need to improve software beyond MS-Dial or other software to really incorporate all these steps. To say, look, I recognize you have the blanks. I recognize you have duplicate injections. Don't worry, I'll take care for it for you.
Right.
And if you didn't have it, I'll give you a big red flag and say, look, you didn't have blanks. You're not my friend. Software should tell that, right?
Yes, it should.
And also like replicates. In our lab, we of course use tons of replicates, but we do not do triplicate analysis of every sample. We don't do that. right? We instead use quality control samples,
Yeah.
in principle with similar ideas behind it, and
Yeah.
we clean our data sets a lot.
Mhm.
You know, so that's, but it's all a little bit like stitched together, you know, as you say.
Mhm. Mhm.
You know, we have MS Flow algorithm. We didn't have a good experience with Cleaner, MS Cleaner. Didn't work in our
Mhm.
head, honestly.
Mhm.
But that's the other thing, like, you know, the software comparisons, the benchmarking. There's a lot to do in the metabolomics field.
Mhm.
And we try to bring it together in the consortia, but that's usually work that's not being funded. You know, you get funded to analyze green tea or cure cancer, or, you know,
Yes.
promise the blue from the sky, but you're not, you're not funded to do the groundwork of, you know, benchmarking, for example. But we
Right.
all agree that this is needed. And so as a community, we come together and, you know, have symposia at least on that topic. And, you know, there are some benchmarking round robin tests going on where we approach this.
Mm.
All right,
Yes.
so that's my 5 cents. You know, to wrap it up in terms of my opinion on all these things. Congratulations again, Nadja, and good luck for your future work. I think the chemosphere of biology is giving us a lot of work, and not only in drug development, but also in just understanding plant and fungi biochemistry, you know, which is interesting. And with that, Arpana usually now has one slide to advance our seminar series.
So our next speaker is Dr. Vega de Cunha. She's coming from the Inherited Metabolic Diseases and Metabolic Pair Group at the De Duve Institute. She's an expert in metabolism and enzymology and has been working on inborn errors of metabolism. So we're really excited to have her. Do keep in mind that it is on a Thursday, not our regular times of Wednesday seminar. So if you have the time, please make sure you register, and we'd love to see you then. And hope you guys all have a great day and great week.
Thank you again, everyone, for attending, and thank you, Professor Cech, for joining us today.
It was such a pleasure. Thank you for having me.